Hello and good evening. So, exam is just round the corner, and I can pretty well understand what is your state right now. You must be very tense, right? Like what to do, what to revise, and all, isn't it? Well, first and foremost, stop panicking. That is the first rule. Don't panic. Because if you start to panic, you will all jumble up the things. Okay, so have a very cool head. I know it's difficult, but at least try to have a very cool head. All right, and you must have made a plan as to what you must do. If you have not, after this webinar, sit and make a plan. Okay, make sure that you don't bring in any new topic right now. Okay, focus more on revision. If you think that, okay, I have revised well, these are the things which I already know, now I can venture into a new topic, then just little small things, but I will suggest more of revision right now, more of question solving, more of revision. Okay, because this is a high time right now. The all the study part is over. Okay, and definitely follow a routine this time. Okay, and don't keep anything new or any kind of revision or something on the day before your exam. Okay, the day before your exam, just be calm, solve some general aptitude questions, go through the pictures. That's it. Don't panic the day before your exam. Okay, so today what we'll do in this powerhouse, in this 2 hour powerhouse, is we will revise some topics from unit 3 and unit 10, and we will look into some questions. So it will be like, maybe I'll give you the question first and then I'll discuss the topic. Okay, discuss that answer, and simultaneously discuss the related topics over there, or I will discuss the topic and then give you the answer. Okay, so, shall we all start? Are we prepared to see how is our revision going? Let's see. All set to go? And I'll start with the first question. Yes, I'm getting some yeses. Yes, here. Right, right. I'll keep checking your comments okay, in the live chat, but let me explain, and then at times I will check. Okay? All right. So the first question is here. So we'll first look into a question and then we will revise. In meantime, when you're reading the question, try to recap those concepts. All right. So here they are saying that following statements were made with respect to transcription in eukaryotes. RNA polymerase 3 synthesizes mRNAs in nucleoplasm. The target promoter for RNA polymerase 3 is usually represented by a bipartite sequence downstream of the transcription start site. The assembly factors TF3A and TF3C assist the binding of the positioning factor TF3B at the precise location. TF3B is the last factor that joins the initiation complex. And lastly, phosphorylated serine residues in the C-terminal domain of RNA pol 2 serve as a binding site for mRNA processing enzyme. So you have to select the correct combination of the statement. Himali, no, you haven't missed anything. We have just started. Okay. Yatin, Ria, Soumya, you have given me the answer. Okay, many of you have given me the answer. Okay, so basically, from this question, you can understand that when you're learning the transcription, you have to focus on the transcription. Obviously, the prokaryotic transcription, if I compare with the eukaryotic transcription, is quite easy, right? But in eukaryotes, there are many things you need to focus on. We mostly do make the mistake of focusing on the transcription carried out by RNA polymerase 2. Okay, but the initiation and the termination part is a bit different in the case of your pol 1 and pol 3 as well. Okay, if you compare it with your polymerase 2, and there are obviously slight differences, but yes, at least the initiation and the termination you should study, because question comes from there as well. Okay, now if I look into this question, see, in the first case they are saying that RNA polymerase 3 synthesizes mRNA in the nucleoplasm. Is it the nucleoplasm? Like, what is the nucleoplasm? You have this cell here, you have the nucleus here, and you have a nucleolar region here, right? So this part here, this is your nucleoplasm. So they are saying that RNA polymerase 3 synthesizes mRNA in the nucleoplasm. Again, another thing. Nucleoplasm, fine. But what about mRNAs? Is it mRNAs that RNA polymerase is synthesizing? These are the things we need to focus on. See, over here in the statement, they have given you RNA polymerase synthesizes mRNA, right, in the nucleoplasm. This particular thing, this particular concept can come as your 2 marks question as well in your part B. Okay, where they have given you the polymerases, the three polymerases, and they are asking you like which one is transcribing which kind of RNA. Okay, and where it is transcribing it. So this is one concept as a whole. All right. Right. So in this case, obviously it's not polymerase 3 which transcribes the mRNA, it is polymerase 2 which transcribes the mRNA, right? Next, they are saying the target promoter for RNA polymerase 3 is usually represented by a sequence downstream of the transcription start site. So for this, you have to understand the initiation of transcription for polymerase 3. To understand statement C also, and to understand statement D also, you need to understand the the initiation part of transcription of pol 3 transcription. Now, if you look at this statement E, all right, in statement E, they are saying that phosphorylated serine residues in the C terminal domain of RNA pol 2 serves as the binding site for mRNA processing enzyme. So now, after they have talked about polymerase 3, they have also brought in polymerase 2. Okay, and C terminal domain CTD, where the serine residue is phosphorylated, we all know about that. Okay, in the initiation of transcription by polymerase 2. But is it acting for the binding site of your mRNA processing enzyme? No, right? The phosphorylated serine, this is doing something else. Okay, we will see what. So here, three different concepts has been amalgamated together. Have you noticed that? Three different concepts. This can come as a separate concept. This can come as a separate concept, and along with that, here is the third one. Okay, so you should study like this way. Focus on each and every concept. So we'll get into the concepts, but for the time being, the correct answer of this is option 2. So those who gave me the answer as B, C, E, it is correct. Okay. So first, let me just discuss about the initiation part of your polymerase 3. So in case of your polymerase 3, what we have here is this polymerase 3. What does it transcribe? It is responsible for transcribing your 5s rRNA and tRNAs, right? So for 5s rRNA and for tRNA, you have two different initiation sites. Okay? Now, देखो, in the case of your if it is a tRNA, all right? Yeah, exactly. So if it is a tRNA, in that case, what we have is we have this A block and the B block. Okay. First and foremost, look at one thing. These are the sequences which have been mentioned, which is downstream of your start site. Okay. So in that case, statement B, which was my statement here, this statement is right, is downstream of the transcription start site. Okay. Now we have these blocks A and B. So these are the sequences. And in this case, we have TF3C, which comes and recognizes this particular sequence. Okay. After that, we have TF2B, which is coming and binding to this TF3C. Okay. Now, so I can also say that TF3C is recruiting TF3B. All right. Now, after that, there are several other binding factors which comes and binds. Like you have your BRF1. Okay, BRF1 you have, which is your binding factor 1. You have other factors like BDP1. Okay, and you also have the TBP, which is your Tata box binding protein. Fine. So after your C has recruited B, all these binding factors comes and binds here. Okay. And then your TF3B recruits the polymerase 3. Fine. Now this is in the case of the promoter for your tRNA. All right. Now if it is your 5S RNA, okay, so there is a second type of promoter for your 5S RNA. In that case, again you have these sequences A block, C block, okay, IE block, which is at the downstream of your start site. If you notice, this is at the downstream. Okay, now in this case we have this first we have this TF3A. Okay, this binds here and then it recruits your TF3B. So first and foremost, TF3C is doing the same thing. It is recognizing the sequence. Then TF3A is coming, which is recruiting your TF3B. Okay, and then TF3B by the same process, it is recruiting the polymerase 3. All right. So these two initiations you must remember. All right. So, accurately initiated transcription by RNA polymerase 3 requires the action of three transcription factors TF3A, B, C. TF3B is central in the sense that it alone suffices for the recruiting RNA polymerase to the promoter for securing repeated rounds of accurately initiated transcription. The large multi-subunit TF3C plays two critical accessory roles in initiation transcription. And what are those? First, it recruits TF3B to genes that lack strong Tata boxes. And secondly, it protects polymerase 3 genes against transcription blocking encroachment by chromatin. Okay. Now, in the third case, what did we see here? We have TF3A, which binds exclusively to the promoter of your 5S rRNA genes and serve as the platform for binding TF3C to these genes. Okay? So this goes with the initiation of your polymerase 3. Now, since we are doing polymerase 3, let us screen is not visible. Okay. Right. So let us revise these parts also. We will also look into the initiation of your polymerase 1 as well. Now, here we have what, as I told you, you need to revise this. So, the different types of polymerases, the location where it is synthesizing, the different types of rRNAs which are being, the different types of RNAs which are being synthesized, okay, by each of the polymerase, And yes, definitely you do get a lot of questions from this alpha amanitin as well. Okay, so whether it is sensitive to this particular polymerase or not, you need to remember that as well. Okay, now every polymerase has got this regulatory sequences. Now this is what I'm focusing for your polymerase 2. In the case of polymerase 2, we have these so we have the start site, okay, for transcription here, and we have the core promoter region, which is comprising of the Tata box, and uh, there are several other initiation sites as well. Now remember one thing here. Tata box is not the only uh, it's not mandatory that the initiation site has to have the Tata box. Okay, this is once there was a question like this, like every promoter site, every core promoter site contains data box. That came as a uh, true or false question. Okay, select the correct answer. So over there, remember data box is not present in all the promoters. Okay, it's not mandatory. There are many other core promoter sites, all right, which can be present here. Now, apart from the core promoter, we have the proximal control elements as well. All right, like we have the GC box, we have the cat box, and what we have here is what's the difference between this proximal control element and the core promoter element is that your polymerase and the transcription factors, they are all going to bind at this position here. All right, and all your activators, the enhancers, and all, they are going to interact with these proximal control elements and other sequences which are also present, like enhancer is one sequence, right? And you have the activators which comes and binds to this proximal control elements as well as to the enhancer sequences. So these small tiny things you need to keep in mind. Now coming over to your initiation of polymerase 1, RNA polymerase 1. So in case of your RNA polymerase 1, we have this core region and uh, we have this UCE region. Okay, so this is your upstream site here, to which this upstream binding factor binds. Okay, so it binds both at your UCE region and it binds to the core sequence here and forms a dimer. Now once your UBF has bound, then it recruits a complex which is known as your SL1 complex. Okay, now when this SL1 complex, this is an initiation factor. Okay, and it contains many other factors here, like we have the Tata box binding proteins, we have the Tata box associated factors, the different factors here. Once this SL1 has got recruited on this particular sequence here, okay, the upstream control elements and the core promoter region, then your polymerase comes and binds to this core site. Okay, so this is about your polymerase 1. Okay, polymerase 1, which is responsible for synthesizing your rRNAs. Okay. Now, as I told you, these control elements, the proximal control elements as you have, or the enhancers, okay, just let us quickly recall what is enhancer. Enhancers, many a times a question comes from enhancer, how it is different from the other sequences. First and foremost, remember that enhancers can be it is not necessary that enhancer has to be very close to the gene. It can be at a distance. Okay, enhancers can be at a distance. Okay, secondly, enhancers are binding toward the activators. All right, thirdly, enhancers can work in both cis and trans manner. Now what is the cis and trans manner? Suppose here you have a DNA. Okay, I'm just drawing a line to for the timing. So this is DNA number 1, and here you have another DNA which is number 2, or rather let me say that you have two genes here. Okay, this is gene number 1, this is gene number 2. Okay, to be more precise. Now say somewhere down the stream here I have an enhancer, okay, which is present on this particular DNA strand. Now this enhancer, if it is controlling this gene 1, then this is known as cis, okay, the same one. If it is controlling the gene on a different DNA, okay, on a different chromosome, then this is known as your trans. So it is only enhancers which are the positive regulators. These are the positive regulatory sequences, okay, which can work in both cis and trans manners. Fine. The core promoter sites, the proximal control elements, they cannot work in the trans manner. They are always cis. Same features there is for the silencers as well. Silencers are just the negative, they're just the negative regulators. Okay. Otherwise, the same features are there for your silencers. So you get a lot of question on the properties of enhancers. To keep in mind about that. Now, let me just quickly come over to this place. What is the function of the activators and all? So, see, first and foremost, in the case of your prokaryotes, we have the sequence here. Activators comes and binds to these sequences. It recruits or it helps in the recruitment of, uh, actually not in prokaryotes, in eukaryotes only I will say. Activators are coming and it is helping in the recruitment of this transcription factor, and then the polymerase is coming, okay. But mainly activators are very, very important when it comes to like unwinding of the chromosome. Remember that activators can regulate the co-activators, okay. Activators and co-activators, they're working together. So in the eukaryotic chromosome, what we have is the entire DNA is spoiled, right? So we need to unwind that DNA. We need to expose the gene, only then the transcription can happen. So for that, the activator comes and binds to the enhancer regions. It recruits the hat, okay, which is your acetyl transferase, which sorry, which places the acetyl groups, and as a result of which this gene this region is unwound, and then we have the other factors which are coming and binding to this DNA, and it is removing the nucleosome from those regions, opening up the gene portion where now the mediator is coming and helping in the proper recruitment of the transcription factors over here, okay. And finally the polymerase is coming and it starts the transcription. So now if I look into the transcription, now if you need to look into the transcription of RNA pol 2 in details. So RNA polymerase 2, you need to look into details. What are the steps in that? We have the start point, okay, at my core promoter region, which is being recognized by your TF2D. So TF2D is coming and binding to the Tata box here. Now this TF2D recruits TF2A and TF2B, okay. Now TF2A, this is holding on to the TF2D, and B helps in the proper positioning of the RNA polymerase. Now someone has to come to bring the RNA polymerase, so that is being done by TF2F. Okay, TF2F next brings the RNA polymerase and it places on the co-promoter side. Then TF2E and TF2H comes, and this stabilizes the entire complex. Now TF2H has got very important role, that is a phosphorylation. The first phosphorylation happens on serine 5, okay, on the CTT tail of this polymerase. There's a CTT tail here. So on the serine 5 of the CTT tail, we have the first phosphorylation. The second phosphorylation happens on serine 2. But remember your H, TF2H, this is only causing the first phosphorylation, okay, which is your serine 5. And this is the end of your initiation step. When we get into the elongation step, then we have other factors, which is your PTEFB, that phosphorylase the second serine on the CTT tail, all right, and that completes the phosphorylation step, ultimately after which the RNA polymerase is all set to move forward and start transcription of all the genes, start transcribing all the genes. ठीक है. So that takes us to the elongation. Now, in the elongation step, what we have to remember here is that as your elongation is taking place, so there are the elongation factors which are all present. Like you have the several factors are there. Like you have the HS PT5, you have the TAF SF1, okay, you have the TF2S. All of these factors are there, which are somewhere or the other helping in the elongation process. Now, particularly if I want to mention about this HSPT5, this is the one which is recruiting the 5 prime capping, okay, which is recruiting the enzymes which are going to put the 5 prime, which is going to put the cap at the 5 prime region. Okay? Now, so from this information, what we understand, again, a portion from where a lot of question has come, that capping and splicing, both of this happens along with elongation. Okay, it is not like after we finish the elongation step then this process is happening. No, it happens simultaneously. Okay, again, this is a very common place where you get questions from. So this is recruiting the capping enzymes, and you have this TAF SF1, which is going to recruit the splicing machinery. Okay, so by the time we are ending with the elongation step, we have an mRNA where all the introns has been removed. Okay, only the exons have been joined, the introns have been removed, but the RNA is still in a hybrid condition with the DNA, and the polymerase 2 is still going on synthesizing the RNA even after we have done with the initial stage. So now that brings us to the termination part. All right. Now termination in eukaryotes, again, since we have 3 polymerases, there are different types of termination. So Pol 1, it contains specific sequences of base pair, 11 base pair long in the humans, 18 base pair in mice. All right. And that is recognized by a termination protein, which is TTF1. This helps in the detachment of the RNA from the DNA. In case of our polymerase 3, we have short stretches of 4 to 7 U's at the 3 prime end, which again helps in the dissociation. And RNA polymerase 2, as we have already seen, we have to study that in details, right? So, in case of Pol 3, what we have here is we have 2 different sequences. We have this AAU AAA sequence here, and we have the UG sequence here. This AAA sequence, okay, this is being recognized by CPSF. Now, CPSF, this is your cleavage and polyadenylation specific factor. UG is recognized by CTSF, sorry, CSTF. Now, this is the cleaving factor, okay? Now, once this CSTF has bound, then this entire thing bends, okay? Because this CPSF and CSTF, these two binds together. So, in that case, this RNA, it bends like this, okay? It forms a loop like this. Now, after that, you have this CTCF, which makes a cut. Okay, can you see here? It has made a cut here, right? Now, at that cut point, you have this PAP enzyme, okay, which is your polyadenylation polymerase, which comes and binds here, and it starts recruiting the A's here. Okay, now this is in a template independent manner in which this A's are being recruited. Again, a very basic question. Okay, from here you can get. So now, so that is being done with the pre-mRNA. Okay, so pre-mRNA is set. It has got a cap here. It has all the introns has been spliced. Everything has been done, and the 3 prime end here. This is also getting protected by this polyadenylation. But what about this Pol 2? This Pol 2 is going on synthesizing, transcribing the mRNA. So in that case, there comes a endonuclease. Okay, now see one thing. Here comes another question. So, what is the importance of the 5 prime cap and the 3 prime cap? Obviously, to protect this pre mRNA from the endonucleases, right? One of the major factors, apart from the fact that it helps in the transportation of this pre mRNA from the nucleus to the cytoplasm where the protein synthesis is going to take place, as well as it helps in the initiation of protein synthesis, okay? Initiation of translation. So this 5 prime cap 3 prime polyadenal tail, it has got three different functions. So in this growing polypeptide, it does not have any such capping, any such protection. So obviously the endonuclease XRN2, this comes and recognizes this region and starts cleaving this portion here until it reaches this polymerase. Okay, so it cleaves off all the entire mRNA, and after which your polymerase dissociates. So this is the termination of your uh, eukaryotes. Okay, now I'm going a bit fast because this is a revision part. I'm not going to the details of it. Okay, so that is the entire eukaryotic transcription. All right, I'm not going to the prokaryotes, but prokaryotes is very, very easy. Okay, the complex part is the eukaryote, and that is the hotspot. Now, I'm not saying that question doesn't come from the prokaryotes, but yes, eukaryotes, there are many basic things which you tend to miss out. Okay, so that's what I'm highlighting here. So let us quickly move to the next question. Let's see who can answer this next question. So many organisms encode only 18 amino acid tRNA synthetase. These organisms lack the amino acid tRNA synthetase that use as one of the substrates for direct amino acylation. Which one of the following statement represents the correct option? So who is going to give me the answer? Okay, I'm giving you the answer here. Any idea? Now, see, here is one question, one concept. That amino acid, how many amino acid tRNA synthetase do we have? Do we have just one? Do we have all 20 for the 20 amino acids? Actually, we have all the 20. Okay, for the 20 amino acids, we have separate amino acid tRNA synthetase. But yes, in lower organisms, instead of 20, you have 18. So they have some other systems of modification. Okay? Okay. So some of you have given the answer. The correct answer is option 3. So let us understand this concept here. So normally what is the job of this amino acid tRNA synthetase? It has got three domains. Okay, one domain where the amino acid is getting being placed. Then we have another domain where the ATP is coming, and the third domain where your tRNA and tRNA with its anticodon is going to come and bind here. Okay, now these three domains, they are very, very specific, very specific. Okay, now what happens here is first step first step is the amino acid and the ATP. Okay, the binding of your amino acid to your ATP, and in the process what is happening, a PPI is getting released. Now comes the specific tRNA. Okay, and mind it that amino acid tRNA synthetic, it has got its own capacity to understand if it is a correct tRNA or not. Okay, because of the binding domain. Fine, so it can check. So then your tRNA comes and binds, and then this amino acid is transferred onto the tRNA, and that is your uh, you know amino acylation of your tRNA taking place. In most of the cases, in most bacteria, generate one of the two amide amino acid tRNAs, ASN tRNA or GLN tRNAs by transamidation of the mischarged aspartate tRNA or glutamate tRNA catalyzed by heterotrimeric aminotransferase. Okay, so what happens here is, first this particular tRNA is getting charged, okay, by aspartate, and then there is transamidation that takes place and it converts to the particular amino acid. Okay, the same happens with your glutamine here. You have the GLN tRNA, okay, where your glutamate is getting is binding, and then again this changes and gives you the correct binding here. Okay, so the process is your transamidation takes place. So let us quickly revise this translation part. So what you first have to understand is the different properties of the genetic code. So we have the very important properties: universality, right? Then we have what? Specificity. So universality is saying that the same codon is going to code for the same amino acid in all the organism. However, there are some exceptions to the rule. Question comes from this part. What are the exceptions? All right, so you better revise this section as well. Specificity is saying that the codon and the amino acid, they are very specific. Okay, so a particular codon always codes for the same amino acid. Then we have, apart from that, we have the degeneracy. Okay, now degeneracy of the code is saying that more than one codon can code for the same amino acid. Like for example, if you see the glycine has got four codons. All right, and in related to degeneracy, definitely read about the Wobble hypothesis. Okay, because it's related that why degeneracy of the code is happening. That is connected to the Wobble hypothesis. So read this together, and of course, the last one, it is non-overlapping. Okay, non-overlapping, that means it doesn't have any comma, it is comma-less. Okay, these are the four very very important properties of your code, genetic code. Apart from that, it has got a start codon and in a termination codon. All those properties are there, but these are the four very important uh, very important principles which are associated. Okay, now, if I look into the initiation part of translation in the case of prokaryotes, it is very simple. All you have to remember from here is the different sites of your mRNA of your ribosome, and yes, with this, definitely study about the different subunits of the ribosome. Okay, the different subunits of the ribosome and what are they made up of? Subunits of ribosome and the composition. For both your prokaryotes and eukaryotes, you get questions from there. Okay, now if I look into the prokaryotes, and we have the small subunit as 30s. Now 30s, what are the things which are present here? You need to understand there is a P site, okay, there is a site, and we also have here the 16s rRNA. Okay, which is going to bind to the shine Delgarno sequence, which is present just at the start of your initiation point. Okay, this is what helps in the recruitment, in the proper binding of the small subunit to the mRNA. All right, otherwise, how to understand that which is my AUG, which is my initiation codon? Okay, AUG can be present anywhere. Okay, now which is the start point, which is the starting AUG, that I need to understand. So that is marked by your shine Delgarno sequence, and you have the 16s rRNA which binds with this shine Delgarno sequence and positions this small subunit to this particular start site. Okay, after which, what we have here now, before this small subunit is binding to the start site, we already have placed IF3 and IF1. So recall the functionality of IF3, IF1 over here. What are they doing? After which we have the IF2 with the GTP. So wherever you have this GTP, ATP, keep in mind, because and what are they doing? Okay, because again, question comes from these small things here. So what we have here is IF2 is coming and it is bringing in the FMET tRNA. The first tRNA is always FMET. Now I'll read a bit about this FMET, like if FMET is kept or if FMET is removed. Now obviously in the processing part, the post translation processing, the FMET is removed from here. Okay. So this is again a small thing. Each and every step can be asked in the question. That is the beauty of your unit 3. Each and every step can be a question. Okay. And obviously when you have all the things set here, then your 50th subunit comes and binds, and that hydrolyzes the GTP associated with your IF2 into GTP, and then all the three factors are removed, and your large subunit binds to the small subunit to start your prokaryotic translation initiation. Okay, this is as simple as that. But what about the eukaryotes? Eukaryotes, there is more things to remember here. Okay, there are many things to remember here. What you have to remember here is there are many factors which are associated with this. So break it up. Okay, break it up. If you haven't studied this yet, study it in the stepwise manner. It will be easy for you. First and foremost, understand how you're forming this 43 pre-initiation complex. Okay, so what are the things which are making up this 43 pre-initiation complex? So here what we have is the 40th subunit. We have the IF3 and we have the IF5, but what we have here is the IF2. Okay, so this IF2 with its GTP and the FMT RNA, this is also known as your ternary complex. Okay, this ternary complex comes and binds with your small subunit, which is already having these two factors at the E and the A point, just like how it was here. And with that, you have the IF3 and you have IF5. Okay, so that makes your 43 as pre-initiation complex. On the other hand, another thing is going on. What is that? It is a formation of this, you know, uh, the heterotrimer complex. EF4F heterotrimer complex. All right. Now, this heterotrimer complex is comprising of 4E, 4G, 4A. Now, see what are the things where they are binding. Like 4E is recognizing the 5' cap. So, I told the 5' cap and the poly A tail that also helps in the initiation of translation. So, here it is. So, it is binding to the 5' cap. You have the poly A binding protein which binds to the poly A tail, and that, these two proteins, they bind together, and this entire mRNA forms a loop. Okay, you also have this 4G which is binding here, and you have the 4A which is responsible with 4G. This is recognizing this PAB poly A binding protein. Okay, and then 4G and 4E, it binds together, and that helps in the looping. Okay, it is a 4G 4E which binds together and helps in the looping of the RNA. Then you have the 4A, which helps in the removal of the secondary structures, and 4B helps or triggers 4A activity. So this is your total of EIF 4F heterotrimer complex. Okay. Now these two things gets associated together and forms this uh, 40S 48S complex, which ultimately scans this entire mRNA and recognizes the start AUG. Now in this case, the starting AUG can be recognized by the sequence here. Okay, Kozak sequence, and then the entire initiation is taking place. So prokaryotes is a bit eukaryotes is a bit complex, so take it stepwise. Now after that, elongation takes place by the elongation factors. Okay, we have the different elongation factors. We have the uh, EFTU, we have the EFTS, we have the EFG. All right. Now TU has got the GTP, which is restored, which uh, which is restored by your TS. Okay, when this GTP gets hydrolyzed to your GDP, then TS can recruit this GTP back on TU. Okay, and then comes EFG, which actually gives a push to the ribosome, which can then move forward. So like that way, the elongation part takes place, and ultimately we reach to the termination end. Now this termination, more or less, it is the same. Okay, okay, I'll just come to this later on. Let me just say something more of this. Now, see, the termination part is a bit same in both prokaryotes and eukaryotes. There are some recognition factors, okay, some release factors which are there. Now, there are different classes of this. There is class one. If I talk about your prokaryotes, we have class one where we have the RF1, RF2. RF1 recognizes your UAG, RF2 recognizes your UGA, and both of them can recognize UAA. Okay? Then we have the class 2 release factor, which has got this RF3. All right. Now, in the case of your eukaryotes under class 1, we have the ERF1, which recognizes all codons. Okay? And then we have the ERF class 2, which has the ERF3. Okay, now ERF3, this is what now class 1, actually I will say class 1 is as you can understand, it is helping in the recognition of the stop codons. Okay, and this class 2, this helps in the release of the polypeptide chain from your tRNAs, after which comes the recycling factor, the ribosome recycling factors, which helps in the removal of the tRNAs, the tRNA without the polypeptide chain, and also separate the two subunit. So if you see here, this is a summary of the entire thing. We have here the A site, which is having the stop codon here. All right, to which we have this class 1 factor coming and binding. Okay, now this class 1 factor and the class 1 factor, this helps in the removal of the polypeptide chain. Okay, so now what we have is, we have the two tRNAs, and we have this class 1 factor. Then we have the RF3 GTP, which comes and binds here. This GTP, now observe this very carefully. Okay, it is not GTP which is coming, it is the GDP which is coming. Okay, now this GDP is converted to GTP, and this releases the class 1 RF factor. Okay, now what happens, this RF3 also is released. Fine. Now comes the recycling factor, ribosome recycling factor, which comes and binds here to this A site, and it dissociates the two large subunit. Okay. Now, what is happening? You have this EFG along with this GTP coming and binding to this recycling factor. Okay, and what this EFG does, it gives a push. Okay, so when it is pushing this particular recycling factor here, then automatically what will happen is, this tRNA will be pushed to the exit site, and from there it will just go out. Right. Now when all the blocks here are cleared, now this subunits can be separated. Now once the subunit has separated, immediately IF3 comes and binds to to this site here, to prevent any large subunit coming and binding. Okay. Now if I talk about the eukaryotes is also almost same. It's almost the same. Okay, so here we have the class 1 and class 2 RF factor binding together, and the same, there's this GTP coming and binding. All right, and it releases the polypeptide chain, and then the GTP hydrolysis release the peptide, and ultimately then it is the same thing. The RFF is coming, and it is releasing the two subunits of this ribosome. Okay? So this is the initiation of the translation to the termination of the translation, after which definitely go through the translational modifications. Okay. So now again, it's time to take up a question. So let's see if you can answer this. Yes, Kruti, I'm getting your messages. Okay. So yes, what will be the answer? Just think about the concept we have just discussed. Okay. We have the small subunit, we have the large subunit. Together it is going to form the 70s. Okay, so if you have more number of 70s, then what is happening now? What the conditions are being changed now, the condition is being changed, and now you're getting more of 30s and more of 50 and less of 70. That means what has happened? Focus on this particular point and tell me. Focus on this particular point. So the correct option is option 2. So protein X is a dissociation factor which functions in the presence of EGF and GTP. Let us understand the question here. They are saying that when ribosome preparation are incubated individually with either elongation factor G or a newly identified protein X or GTP, the profile remained unchanged. So you are getting the same profile. That means what? You are having more of ribosomes which are in the combined form. Okay, not in the subunits. They have not separated into the subunits, but in the combined form. Okay, so likewise, no changes were seen in the profile when the ribosomal preparation was incubated with EGF plus GTP or protein X with GTP. Okay, but when ribosomal preparation was incubated with protein X, EGF, G, and GTP together, then the dissociation happened, because of which you are getting less number of 70s and more number of 30 and 50. That means what? This EGF we already know. Okay, EGF we already know, this is an elongation factor. Fine. And this EGF comes with its GTP. Okay, but what about protein X? What is the nature of the protein X? It's very clear from this second condition here, from this condition, that protein X is what is helping in the dissociation of the ribosomal subunits. Okay, so it is a dissociation factor. See, when you get the question, no, you know the concept. You know the concept. You just have to apply the concept. Okay. So for that, read the statement very carefully and the conditions very carefully. Okay. Now here is another question. So this is more of like understanding. This is more of a statement. Okay, mitochondrial protein synthesis is of prokaryotic origin. Following statements are being made about the ribosome from bacteria and mitochondria. So you have the three statements, which is the correct one. So which is the correct one? For this, you have to know, information. So I'm giving you an information. This is something which I'm not explaining. The correct option in this case is option 1. So statement A and statement B are correct. Okay, statement A and statement B are correct. Bacterial ribosome consists of small and large subunits of 30S and 50S respectively, whereas mitochondria of mammals, these subunits are of 28S and 39S. In bacterial ribosomes, the RNA is to protein ratio is about 2 is to 1, whereas in the mitochondria ribosome, the ratio is usually 1 is to 2. Okay, so here is your takeaway. You can take a screenshot of this, keep this data in your hand. Okay, the difference between the uh, bacterial mitochondria, mitochondria and the bacterial ribosomes. Okay. So changing and coming to the last question from your unit 3. So this is from the last section of your unit 3, where you have the gene regulation in the prokaryotes, and where we have to study about the C1 gene, C2 gene products, crow, okay, C3, and basically we have to study the mechanism in which the lambda phage is going to shift from lytic to lysogenic cycle and vice versa, and when it is going to do so. Okay, that is what is important. So let's do something here. I know many of you have left out this part, thinking it to be very difficult. I will just touch in the basics of it, okay? And then I'll come back to this question. All right. So here is the arrangement. Okay, here is the arrangement of the region which is going to regulate whether it is going to go towards a lysogenic development or whether it is going to go towards a lytic development. Okay, so what we have here is we have the N here, which is an anti-terminator. We have the C1 protein, which has got this two promoter sites, PRM, PR. It has got the CRO, it has got C2, O, P, Q, and the late genes are there. Now here we have the left promoter region. This is the right promoter. This is the left promoter, where we have the PLOR. OL is the operator site. OR is the operator site here. Okay. Now see the basic factor which is determining this shift is whether we have the good condition for growth or not. Okay, now if there is a good condition of growth, then we have this protease FTSH, okay, which is a protease. This is its concentration increases, okay, or its amount increases, and if this increases, then it will block C2. Now C2 is very important to stabilize C3 one. Okay, C2 along with C1 stabilizes. So pardon me, I'm repeating once again. C2 along with C3 stabilizes C1. Okay, so if you block C2, then obviously C1 will not be stable. in that case you will not be able to go into the lysogenic cycle, because C1 is required for lysogen. Okay, so in that case lysogen is not possible, we will go for lytic. Okay, because C1 is not there, so alternatively what you have here is the crow that will be active. Okay, and crow causes the lytic cycle. It promotes the lytic cycle. Now if conditions are bad, then obviously what will happen, this FTSH will be low. In that case you will have C2. All right, C2 along with C3 will stabilize C1. When C1 is stable, it will promote the lysogenic cycle. It will block CRO1. Okay, it will block CRO and promote the lysogenic cycle. This is a very fundamental thing that you have to keep in mind when you're starting to study this regulation. Okay, now here what happens. So as I said here, what we have at the start, at the very start of your infection stage, we have both this left promoter and the right promoter which are active. Okay. Now left promoter controls all the genes which are in the left hand side. Right promoter controls all the genes which are at the right hand side. Okay. So at the same time, what is happening at this time, your C2 is getting transcribed and here C3 is also getting transcribed. Sorry, C3 is getting transcribed here on the left and C2 is getting transcribed here. Okay. Now the C2 which is present, okay, whether it is going to bind to C3 and promote C1 again depends upon the good or the bad conditions. Good or the bad conditions. Okay. Because why? Here there is a competition between the FTSH and C2. Okay, which is going to bind to C3, which is going to bind to C3, and that is how the C2 concentration it is varied. Okay. Now if suppose the condition is good, okay, then what we have here, C2 is active, C2 goes and binds with C3, and the little bit amount of C1 that is being produced from the right hand side promoter, it is going and binding to the C1, okay, and thus stabilizing the C1. Now the C1 will have, the C1 has its own regulator, okay, okay, which is a PRM, which is a PRM. Okay, it will bind to this promoter site and it will cause further production of C1. And this is what is the first step towards lysogen. Why? Because when there is more amount of C1, what it does, it goes and blocks uh, the crow activation. Okay, crow genes is blocked. Okay, in the other on the other hand, C2 which was produced, C2 not only uh, stabilizes C1 but it also goes and triggers the PI here, okay, which causes the transcription of the integrase enzyme, and that helps in the integration of the phage into the bacterial chromosome. Fine. So on one way C1 is blocking CRO, on the other way integrase is being synthesized. So it's a complete channelizing towards your lysogenic condition. Okay. Now C1, remember, there are several properties of the C1. Okay. C1 on one end it is blocking CRO. C1 can promote its own synthesis, so more and more of C1 is being produced. But at the same time, C1 can auto-regulate itself. Okay, it can auto-regulate itself. So after reaching a threshold, C1 will automatically stop. It will stop its own production. Fine. Now see, now, when it is stopping its production, it is in the stable condition. Okay, it is in the lysogenic condition. Now, the external environment is again going to determine whether it is going to come out of the lysogenic condition and get into the lytic condition. Okay. Now, the normal tendency is to come out of the lysogenic condition, because obviously that is creating a stress. Okay, there is a foreign DNA which is integrated into the bacterial DNA, so obviously it is a stress condition, right? So what happens when the stress condition, like how we have read about the SOS, okay, here also we have these uh, these proteins are there, okay, which can uh, degrade this C1. All right. So under stress, we have this. We have the rec A. We have the lex A. Okay, which is switched on, and that degrades the C1. Now, when C1 is degraded, okay, because here this C1, it mimics the lex A. All right. Now, lex A can be cleaved by rec A. So, whenever the uh, condition is a stressed one, then rec A can go and cleave the C1 because C1 resembles the lex A protein. Okay. Now, what is happening as C1 is getting depleted, the amount of it, then obviously this blockage, okay, this inhibition over the crow, that is being removed. Now, when that is being removed, then obviously now crow is getting active. Okay. Now, crow, when it is getting active, obviously it is transcribing all these proteins here. Now, what the crow does first is the crow on the very first, it will go and block the C1. Okay, the crow at the first thing that it does is it will go and block C1. Okay, at the same time, it will promote the transcription of all these Q genes and the late genes. Now, the late genes, they are mainly involved in the formation of the the capsid, the tail, and the head region, the tail region, the entire structure. So what is happening? So on one end, it is the bacteriophage is all ready to produce a new bacteriophage. Okay, resynthesizing the head, tail, and everything. It is blocking C1 to stop moving back into the lysogenic condition. And the third thing it does, it also helps. Actually, it directly does not help, but uh, the binding of the polymerases, the at the promoter sites and all, this somehow blocks this integrase. Okay, so in this case, what is happening here, an mRNA is produced. Okay, the mRNA which has got a CIP region, it has got this integrase and it has got the cis. Okay, just before in int you have a cis. Okay, here you have the cis gene. Now, cis gene is for excision. Okay, coming out from the lysogenic condition, you need the cis gene. So, in this condition, what is happening? The cis, int, CIP, all of them are getting transcribed. Okay, so the mRNA is being formed, but what is happening? The CIP region is being recognized by a RNase 3. All right, and that cleaves this CIP region. Along with the SIB region, this INT also gets cleaved. Okay, so what you're left with in this mRNA is only the cis, which will help in the transition from the lysogenic to the lytic cycle because it will bring out the bacteriophage from the integrated condition. Okay, so these are the things which happens in the next round when the transition is taking place. Okay, so here you can take away one thing first, that is obviously you have to understand the entire mechanism, keep it in part. Okay, try to do it yourself in a flowchart manner, you will remember it in part, like stepwise you definitely do that. And the role of each and every, like role of crow, role of C1, role of C2, these are very very important. Okay, so remember the roles of it. And yes, one more thing remember here is the what happens when int is there and when what happens to cis. So basically, when int is greater than cis, it will cause lysogeny. When int is less than cis, it will cause lytic. When int is equal to cis, it will cause the lytic excision. Okay. Now, to understand this, there are many other small things that you need to go through. So, those who have already read this part, you will know about this. Okay. So, this is the one in very brief. Now I'll go back to the question. So now can you answer? Now can you answer the question here? So they have given some statements. Competition between C1 and C2 gene product determines the establishment of lysogeny versus lysis. C1 binds OR1 first, while CRO binds to OR3 first. CRO binding to OR represses C1 bind transcription. Sorry. So I hope you have recollected this entire thing now. Okay, Soumya is saying option 1. Okay, many of you are saying option A and B. Some of you are saying option B and C also. Option 2. In this case, the correct option is option 1. Okay, first and foremost, there is no competition. This is very wrong. There is no competition between C1 and C2. No. C1, C2 only stabilizes C1. Okay, C2 stabilizes C1. There is no competition between here. What is the competition is there between C2 and FTSH? Okay, for binding to your C3, there you have a competition, but not between C1 and C2. This is wrong. So that is where here B1 is correct, B statement is correct, C statement is correct, and D is wrong is because rich medium favors lytic cycle because C2 is protected from cellular proteases. All right. Now, as I said, this is wrong. Rich medium will cause lytic cycle, but not because C2 is protected. C2 is not protected from the cellular proteases. C2 gets degraded, and that is why you go into the lytic cycle. Understood, everyone? Okay. So this is about the last part of your unit 3. Now let's shift into unit 10. And the first question of unit 10 is in front of you. So as per IUCN, what is the definition of endangered species? Very important. I've given this in class as well, like how to differentiate between endangered species, how to differentiate between your this critically endangered, endangered. Okay. So what are the criterias? What are the criterias that you have to keep in mind? So in this case, the correct option is option 4. Okay, 75% reduction in population size in less than 500 kilometers square area of occupancy, at least 20% estimated extinction rate in 5 generations. Okay, so this is the entire list of your uh, this particular IUCN data here. All right. Sorry, I just skipped. Yeah, this part here you have. And endangered is what? It is a designation applied to species that possesses very high risks of extinction as a result of rapid population declines of 50 to more than 70% over the previous 10 years or 3 generations. A current population size of fewer than 250 individuals or uh, other factors. Okay, like that way you have the vulnerable as well. So vulnerable also you have to study. You have to study the critically endangered, endangered. These are very nearby. Okay, like these values you have to keep in mind. Okay. Now moving to the next question. So this is a question from your population ecology. Those who have attended my class, you should be able to answer this. See what they are asking. At what point expect their population to attain the highest growth? They are not saying that. Be very careful. They are not saying what will be the maximum yield. Okay, what will be the maximum yield at that point. They are saying at what point of time you will get the maximum population. Two are different questions. Two are different questions. I've mentioned this in class as well. Okay, so be careful in what they are asking. So can I get some answers? Yes, Shweta, Kavya, Vishnu, you're all giving me the answers. Just excuse me a minute. Okay, Shweta, Kavya, Kirtana, very good. Anupam, yes. So the correct answer is option 2. Why is it option? Why is it option 2? Because we have talked about the carrying capacity, isn't it? So the logistic uh, growth, it always follows a sigmoid curve, right? Where initially there is a lag phase, there is a log phase, and then there is an exponential growth, after which once it is reaching or nearing the carrying capacity, we are leveling off. Okay, now this is a logistic population when we are taking n and t. But if I take dn by dt by n, then we have the graph as your hyperbola. Okay, now I have told you that in this case, when you have the population at its half carrying capacity, okay, that is the time you are supposed to get the maximum yield. Okay, so at what point of time you will get more number of individual is half of its carrying capacity. So in this case what they have given here is 520 is the size, right, after which the population becomes stable. That means 520 is the carrying capacity of this entire of this region. Okay, so the half of your carrying capacity will be how much? 260. 260 is the half of your carrying capacity. Yes, Kavya. Okay. So that is what you have to keep in mind uh, here. Okay, you have to keep it in mind here that what is your carrying capacity. Okay, what is the total yield, the maximum yield, when will you get it, the different types of curves. This is what you have to keep in mind, because you can be asked about these curves. Okay, they can give you these curves and they can ask you like which is exponential growth, which is for the logistic growth. Okay, because see what is changing here are the parameters. Okay, the x and y axis. If you change the x and y axis, you the same thing you are getting as different curves. Okay, different graphs can be generated. So this is something you have to memorize. Okay, along with the equation you have to memorize like which are coming under your exponential growth, which are coming under your logistic growth, because this is a very um, common question, I will say. Okay. Yes, Anusha, what I said here is, see, in this time there is an exponential growth, right? There's an exponential growth here, after which the you are leveling off. Okay, you're reaching the carrying capacity. So they're saying that at what point of time you're going to get the population growth will be that highest, at what point of time. Okay, what will be the population at that time when the growth is highest? That means they're asking about this particular point. This is the highest point, after which there is a decline. Okay, so this is what, this is half of your carrying capacity. Okay, this is k by 2. So 520, what did they give you? 520, half of 520 is 260. Okay, Anusha. ठीक है. So now let us quickly revise this population ecology part, because this is important. So what are the things that you need to remember from here is definitely the Lincoln Peterson method of estimating the population size. Okay, which is saying that n is equals to mc by r, when n equals to estimate of the population, total population size. M is equals to total number of animals captured and marked on the first visit. C is the total number of animals captured on the second visit, and r is the number of animals captured on the first visit that were then recaptured on the second visit. Okay, then you definitely have to remember this different types of dispersion. Okay, whether it is clump, whether it is uniform, whether it is random, this is something you have to remember, why it is so, and what will be the different values of v by m. Okay, what will be the different values of v by m here, that you have to remember from this part, because you get questions from this v by m values, whether it is greater than 1, whether it is less than 1. Okay, when it is equal to 1, what are the different types of uh, conditions you get from here. And yes, you also get questions from these graphs of the population. Okay, where you have the expanding population, how does it represent? If it is if it is a stationary phase, how does it represent? If it is a contracting, then how does it represent? So here we have the triangular shapes. Okay, we have the urn shape here. Okay, umbrella shape, we have the urn shaped. So the question comes from these shapes as well. Okay, they can give you that when will you get the urn shape kind of graph. Okay, so you have to understand that it is in the stage 4 condition that we are going to get it. When you're going to get a triangular graph, so it is in the expanding condition that we'll get it. Okay. Then about this rapid fall in each upward age group, whether it is stationary, whether it is contracting. Okay, different ways question comes from this section, right? And yes, apart from that, do revise your life tables as well, the calculations which has been taught. Okay, that calculations definitely go through it. And the survivorship curve also, which is in related to that graph, I mean the life table. Okay, the r0 values, how to calculate the r0 values, all those things which I have discussed. Okay, the r0, how to, what does it mean if it is if r0 is greater than 1, if r0 is less than 1, what does it mean, what does it imply. Okay, what does it imply? Those things you have to uh, revise. Survivorship, what is trade-off, what is r and k selection. Okay, the comparison between r and k selection. These are the stuff that you have to revise, because this is a place where a lot of questions do come. Okay, moving to the next one. This is about your diversity. Let's see. This is talking about the alpha diversity, the beta diversity. So I'll just clarify this thing. This large box here, this larger box here, this is telling you a region. Okay, this is giving you the idea of a region, and these smaller boxes here, this is giving the idea of habitats. So I hope you remember what I told you about the habitats, environment, habitats, niche. What are the differences? Okay, what's the difference between them? I hope you remember. So now from here they're asking a question about the diversity. So I'll do something. I will first go through this idea and then I'll come back to the question. Okay? Done? I'll just discuss the idea first and then I'll come back to the question. So first and foremost, see, this is a landscape. Okay, this is a larger landscape, what we are calling it as region in that question. And these are your site A, site B, what has been shown. These are the habitats which has been indicated in the questions. Okay, the smaller boxes. Now, in this case, if you consider this particular habitat here or the site A, how many different types of organism you have? One, two, three, four, five, six, right? Six different types. So when I'm just counting a particular region, this is my alpha diversity. Okay. When I'm comparing between several regions, okay, then that becomes my beta diversity. Like, what is the diversity change between diversity change, okay, between this habitat and this habitat? That will be my beta diversity. When I will compare the diversity change over the regions, over several landscapes, then that will become my gamma diversity. Okay, so B alpha is for a very small place, beta is for the comparison between two different habitats, gamma is comparison between the regions. So alpha diversity measures the diversity of a particular area or an ecosystem, usually by expressing the species richness or the number of species. Okay, now species richness matlab the different numbers of species which are there. Okay, beta diversity measures the diversity between ecosystems, so between regions, between habitats. Sorry, not between regions, between habitats, or between the ecosystems. Okay, that is your beta diversity. Okay, and what we are measuring here is the species change. Species change we are measuring. Okay, between the ecosystems. And what is the gamma? Gamma is the largest one, the vast one, that is measuring the overall diversity across a large geographic region. Okay, now if you have all noted this, I'll go back to the question now. So now let's see if you can answer this. Come on, apply the concept that we have just discussed. You're asked to say which regions shows maximum alpha and beta diversity. Okay, many of you are saying option 1. Let us understand. Let us understand this. See, in this region, we have S1, S2, S1, S2, S1, S2. So it is the same species in every habitat we have. Okay, right? So it's the same alpha diversity here, 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 and here, the same alpha diversity. And if I compare the beta, then it is also the same because we have the same values. Okay, so obviously P cannot be the maximum diversity. Let us talk about Q. In the case of Q, for this we have S1, S2, S3, three species. Okay, here also have the three species. Here also have three species. Here also have three species. So alpha diversity is same in all of the four habitats, right? Now, if I compare the Q, so the condition of Q and P is almost the same, except for the fact that in P there were only two species. In Q there are three species, right? So automatically Q is more diverse than P. The Q region is more diverse than P region. Okay. Now if you come to R, it was only comprising of one species and two species here. Okay. So alpha diversity is also very low. The beta diversity is also very low. All right. And if I consider S, in S I have S1, S2. So alpha diversity wise, this is having two species. Over here there is only one species, but I have a different species, S3. Here also I have another species, but it is a different species, S4. Here also I have one species, but a different species, S5. So accordingly this is also very diverse, right? The alpha diversity, if I compare the alpha diversity, then this habitat is more diverse. Okay, but if I compare the beta diversity between the regions, then obviously your Q and S is the most diverse. Okay, alpha diversity wise, you have the Q which is more diverse. Beta diversity wise, you have the S. Okay, so very good. Everyone has given me the right answer. So far, everyone has given me the right answer. Good. Very good. So now moving on to another concept. Species richness we have talked about. Species richness is the different types of or the different numbers of species that is there in a particular habitat. Okay, the different types of species. Now, how many numbers of individuals of each species you have, that is talking about the abundance. Like species 1, there are 80 in number. Okay, species 2, there are 25 individuals or 25 in number. Okay, so this is talking about the abundance. Fine. This is talking about the abundance. So from here, what you have to find out is the order of diversity. So from here, you can obviously sit and calculate, okay, by using one of the indexes, or you can apply the smarter way. Okay, the three habitats has been given to you. Remember one thing: diversity. Diversity is controlled by species richness and species abundance, both. Okay, because species diversity is talking about a community where there is a variety of organisms which make up the community. Okay, so it has got the species richness and actually not exactly abundance but rather relative abundance. Okay, the relative abundance, I should be more precise. So you know what is richness, you know about the abundance. Now see, Only richness and abundance is not important. There is something else which is important, that is evenness. Okay, evenness is what? It is a measure of the relative abundances of the individual species. Okay, like if I have species 1 2 3, and here I have 10 number of individuals, I have here 11 number, and here I have 10 numbers, so it is almost evenly distributed, right? The three species. So that is evenness, how evenly they are distributed. Okay, which is mainly focusing on the relative abundances of the species. Okay, so evenness should be more for the community to be more diverse. Okay, that means there should not be equal distribution of the species within the community. Fine. So based on this information, how can you find out which is having more diversity, which is having less diversity? Can you find out? This is a shortest method. You can definitely go and sit and calculate, but obviously that will take a lot of time. So you have to know the smartest way. Net is not all about just reading and just going to the exam and giving the exam. No, you have to use a smart techniques also over there to reduce your time. This time is very limited. Okay, so obviously you're not going to sit and compare. You're not going to calculate the different habitats, but rather go for this shorter technique. So now you tell me which should be the right answer. Okay, many of you are giving me the answer correctly. So here the correct answer is option 4. Okay, in decreasing order of the diversity. Decreasing order. So now let's compare this. If you compare habitat Q and habitat R, okay, first let me talk about habitat Q. In habitat Q, see, almost it is 20, 20, 20, all 20, and then again it's all 15. Okay, so habitat Q is obviously the most, is the least diverse, right? Because here there is more evenness. There should not be more even. To for a region to become more diverse, there should not be more. There should not be uh, evenness. Okay, so this is less diverse. If you take R, in case of R, what you have is there is evenness in the species 6, 7, and 8. Okay, or if I can take in 5 also, 5, 6, 7, 8. But apart from that, the other species, look here, 80, 25, 15, 10. Okay, so I can't say that habitat R is absolutely even. No. Okay, there is a lot of diversity here, at least much greater than your habitat Q. So least will be your habitat Q. Sorry. So sorry, I just made a miss. I'll just rectify this. So, like as I said before, evenness. Evenness, it should be more even. Please rectify. More even. Okay. The more evenness is there, the more diverse it is. Okay, more diverse it is. Okay. So in that case, habitat Q is the highest actually. Habitat Q is the highest. Okay, more even, more diverse. uh, in the case of habitat R, there is evenness here, but again there is unevenness here. So next will be your habitat R. But if you compare your habitat P, in the case of habitat P, it 120 followed by 25, 1. Okay, so automatically this is the highest, having the highest diversity. You can also do it by calculation and get the answers, but you will come to the same answer, same conclusion. Okay. Yes, q has more evenness. Q has more evenness, so that means it is more diverse. Okay, it is more diverse. Okay, there is more relative abundance. So what you have to revise from this section is definitely the different indexes that you have: Shannon diversity index, then Simpson's diversity index, how to calculate, how to do the sums, because you get direct questions from this Shannon index, you get direct questions from the Simpson's index. Okay, calculation where you can't use the shortcut, you have to go for the calculation. Okay, so it's better to revise these indexes as well. And definitely revise the important definitely revise the important organisms here. Okay, so like you have the ketone species, you have the dominant species, you have the rare species, you have the common species. Okay. So, what is a keystone species? Remember, keystone species, they play a very important role in controlling the overall food chain. Okay, but they need not be in. They did not be having a larger biomass, or they did not be in have they did not have a very high abundance. Okay, is their role it is their role that they have is automatically controlling the entire food chain. Okay, if you remove the keystone species, then the entire food chain gets disrupted. Okay, but the dominant species, on the other hand, that is also important. That is also controlling your food chain. Okay, the environment, the community of that area. But most of the time, these dominant species, their biomass is also very large. Okay, and that is how they are controlling that diversity of that area. Like, for example, if you consider this tree here, okay, this is a larger tree. Okay, that means it is having it is creating so much of shadow. Now, when you are creating a shadow here, that means what? All the shade loving plants will be able to grow over here near this tree. All the animals or the organisms which like to stay in the shade, under the shade, they will be able to grow over here, right? So you are enriching this environment, you're creating more diversity in this environment because of your biomass. Okay, so that is how you are a dominant species. So if this species is removed, then obviously this entire community which has developed under this tree, okay, that will have a huge impact. Okay, now next we have the bottom up and top down. Again, a place from where you get a lot of questions. Okay, bottom up and top down, how you are controlling it. So like bottom up means you are doing it from the plant side. Okay, so what you have here is, if you decrease these nutrients, then automatically there will be less number of plants. Okay, that is the producers. If there is less number of producers, then obviously there will be less number of your herbivores. Okay, if less number of herbivores are there, then there will be less number of your carnivores, and so on and so forth. Okay, so this is your bottom up. Okay, as you are going from the bottom, the entire food chain, it is uh, getting impacted. Okay, having a huge impact. But if you come from the top down, like if you say, suppose if you increase the carnivores, okay, what will happen? The herbivores will decrease. All right, if herbivores are decreased, that means what? The plants will increase. So can you see there is an alternative impact here that can be seen if it is a top down control. Okay, so this type of control is also used to control the pollution of a water body. Okay, to control the algal bloom, you use this kind of top down model where you are going to reduce the carnivores, you're going to reduce the carnivores. Okay, that will increase the herbivores. So if the herbivores are increased, then obviously the planktons, which is your algae and all in that plant body, they will reduce. So that is how you can, control the algal bloom as well. Yes, sea otters are one example of the keystone species. Okay, so this is again another very another place where you get a lot of questions from here. Now, apart from this, definitely go through your the stratification, okay, and about the community, revise what is resilience, what is redundancy, okay, the graphs related to redundancy, the graphs related to the resilience, okay, the idiosyncratic manner, that means the community stability. Basically the heading is community stability, okay, that also you should revise. And with that, the different cycles, okay, you have the carbon cycle, nitrogen cycles, revise those cycles as well. And that brings us to the last question of this session of today, the last unit 10 question. So let's see who can answer this. So in this case, they are showing you energy flow within a single trophic level where I is the amount ingested. Please make a note of these. Okay, I is the amount of ingested, Na is the amount of not assimilated, R is the respiration, and Pn is the biomass production. Okay, so from here they are saying if one of the following options represent the correct value for Pn, Na, R, and I in kilocalories respectively, if Pn minus 1 is 1000 kilocals, Pn minus 1 is 20%, A by 1 is 35%, and Pn by A is 20%. I'm giving you time for this because calculation is required here. Okay, Sandip is saying option 3. Okay, have you done the calculations? Shall we discuss? Are you still doing the calculations? Okay. Let us do this. Please excuse me. So we have Pn minus 1 is equals to 1000 kilocals, right? We have I by Pn minus 1 is equals to 20%. That means what? I receives 20% of 1000, which is equals to 200. Okay, 200 kilocals this will be. Next I have A by I. A by I is given as 35%. Okay, now 35% of what is I? That is your 200. So 35% of 200 means it is 70 kilocals. Very simple calculation. Fine. Now, so what is your basically A by I is given as your 35%. That means A receives 35% of I. Okay. So that means A is equals to what? It is your 70 kilocals. Okay. Now what is Na? Na is equals to I. See, Na is what? I minus A. Isn't it? I minus A. This is equals to Na. So that means this is your 200 minus 70, which is equals to 130 kilocal. Okay. Now Pn by A, this is equals to 20%. This is equals to 20%. That is, what can I write? Pn is equals to 20% of 70, which is equals to 14 kilocal. Okay. Now what is R? What is R from here? This is your P A minus Pn. We are moving upward. Okay. So your R is equals to A minus Pn. So A minus Pn matlab it is 70 minus 14, which equals to 56 kilocal. Alright. So the correct option in this case will be option number 2. You have to mind it. Now look here very carefully. They are asking you Pn, Na, R and I in this order they are asking. Okay. So don't jumble up the values. You will go wrong otherwise. Okay, so what you have to do is you just have to link this data here with the diagram. So even if you don't know this concept, okay, the biomass production, at what stage, what is happening, okay, then also you will be able to solve these questions. So don't freak out when you get these kinds of questions. Don't freak out, okay? That's a very, very important point. When you get a question, just read through it and see if you can answer. Okay. Now you know what the best part of this computer based examination is that you can always go back to your previous question and change your answer, or you can definitely mark it as review. Okay, if you have answered, you can mark it as review and again go back and analyze it, which was not possible in the case of your OMR sheets. But once you have encircled it, that means it's done. Whether it's right or wrong, you can't change the answer. But in computer based analysis, you can definitely go and change the answer. Okay, till the point you submit your exam, till the point of that. So don't freak out. Try that question. If you can do it logically, try it. There can be mainly three types of questions it comes. One is a very straightforward question, which is coming from whatever you're reading. From there, you get the questions. You get another type of questions where you have to apply, analyze the application. What you have read, you know it. That knowledge you have to apply. Okay. Another type of question which do comes at times is where maybe you don't know the concept. Okay, you don't know the logic behind it, but it's a very straightforward kind of. You just have to use your common sense to solve that answer. Like this one. Okay, this is simple calculation. So those who are good in calculation, those who have that analysis of calculation, you will be able to solve this. Okay, so when in part B also, in part C also, you get some questions like this. Okay, not always regarding the calculation, not based on calculation. It can be sometimes match the column. You will see that if you can match three answers, or if you can match two answers, the rest are already fitting in. Okay, so when you get the question, just read it. Just read it. If you can answer immediately, like if it is a direct question, then obviously you will be able to answer immediately. If it is an analytical question, you have to think. Okay, think about it a bit. And if it is a common sense wala question, then you need to come back and rethink once again. All right, so with this, I come to the end of this session here. So in the end, I want to wish you all the best for your exam. And just first and foremost, be calm. Okay, if you be calm, you will be able to solve it. You have prepared yourself. So have faith in you. Don't panic about the things you have left. No need. You don't even know where the question is going to come from, those areas or not. So what you have in your basket, just have a very strong hold on that. Okay, remember one thing, for net you don't have to you don't have to be thorough with all the topics. We always keep telling this in class, in the webinars. There are few topics, very a few topics which you have to have a very strong hold on. You have already studied it, you have it in your basket. So just be confident about it. Okay, and give your best shot. Just give your best shot. So with that, uh, I come to the end of this, and thank you everyone for attending. I hope this was helpful to you. We have revised Unit 3 sections. We have already also revised Unit 10 in this very short time. Okay, so a part of your revision is already done in these series. So with that, good night.